So here's a quick tour of the cell. These should all be things that you should remember back previously, but we're going to go through it real quickly. It shouldn't take too long. First off, a couple of things you do need to remember though, is that the cell size does help out with how well the cell functions. So all cells can only be a certain size, and they're limited basically by two things. First off, the exchange of materials in and out of the cell, and then also distributing materials within the cell. And that's what this graph is showing. Now basically, if you look at this table here, the total surface area is actually way larger if we're using very small, very individual cubes. If we have a surface area here, a 1 by 1 by 1 by 1, the surface area is 6. Whereas here the surface area is only 150, and we go all the way up to here being 750. But if you look at the volume, and remember the surface area is how much space is on the outside, if you look at the total volume, the volume is the same, even though the surface area is way greater if we use a bunch of little small cells, or little small cubes. And so this is basically the idea with your body. An organism can only be so big, especially a single-cell organism can only be so big, because if this surface area has to be protected, it's kind of like, let's say you have a large property. So you're back in the Middle Ages, and you own this whole barony of area. Well, if you have a barony of area, which is basically you're a baron, and you control this entire area, if you have one large area and you don't have anyone else, and you don't divide into smaller sections, there's a good chance that you're going to miss out on taxes, you're going to miss out on laws being broken, all kinds of things. So it's the same thing with the cell. If the cell ends up being too big, it can't control its area nearly as well, it can't protect it, it can't bring materials in and out nearly as well because there's this gigantic fence. So if you see right here, the surface to volume ratio it's the same as if we only had one cube, and it gets very small if we have one gigantic cube. So remember, it's limited by two things. Now, I always use the fence example of trying to protect your area. If you have a large area, you want to break it down to smaller checkpoints, and that way you can help bring things in and out. And then also, while you're in that larger area, if it's too big, you can't. It takes a long time to send messages across. So I'll kind of show you that when we're in class. So here's just a list of organelles you should know. Again, these are all on the vocabulary list. Like I said, these are the ones that you should know. So what I would suggest mostly doing is making sure you know these cells, that you can find these organelles on the thing, and know key ideas of them. And the best way I always have always seen it is related to something like a city. Okay. Let's say that you have this city, and this city has a nice wooden fence around it. Well, that wooden fence will let some things in and some things out, but not a whole lot. It keeps it pretty strong. Well, that's the membrane. But on the outside of that city, there's this very strong stone wall. Well, that's the cell wall, and that's mostly just there for support and for structure, but the wooden fence is really what controls what comes in and out. In the middle of the city, there's a nucleus, a city hall, and the city hall is where it controls all the blueprints to make all the food, all the proteins. Inside the city hall is a special room called nucleolus. The nucleolus is actually where the blueprints are kept. So again, we're making these proteins. Well, how do we make the proteins? Well, there's a long series of streets right here, and those long series of streets, on parts of them, there's little shops, and those shops, if it's a cell, those shops is where the ribosomes are attached. And so this would be the rough ER and the smooth ER, and it's a channel form. And also these shops are also dotted throughout the entire city. So again, these are the ribosomes, and this is where the shops are present, and they make proteins. Now, also in this cell, we have to have power, and the power in the cell are called the mitochondria. And again, you need to remember the mitochondria are double membranes. And if you remind me in class, I'll explain to you why we think these are actually prokaryotes that have been taken in by eukaryotes. And so we have the mitochondria, and then also with the mitochondria, we have things that are called vacuoles. And basically, vacuoles are essentially the kind of transport areas. If we need to send something out, these vacuoles will go out of the city. Well, where do we package these vacuoles? In something called a Golgi apparatus. And then Golgi apparatus kind of looks this big blobbish looking thing right here. And a Golgi apparatus, it's like the post office box. It gets these vacuoles together, it packs out the proteins we're sending out, and it sends them out. Trying to think anything else that we know we would have. Anything else? So in our city, we also have these cytoskeletons. The cytoskeletons are there for the support structures of the roads. The chloroplasts are specifically in our plant cells. Centrioles are there to help with the mitosis process. Chromatin is basically chromosomes, but they're not wound up yet. The cytoplasm is the kind of the fluid that actually goes through our city to help kind of transport and maintain a certain homeostasis. And these peroxisomes and lysosomes are basically like the garbage men. So again, if you relate it to something you already know, it should be kind of easier. Real quick reminder about microscopes. There's basically different types of microscopes. Microscopes are there to help magnify and to bring up resolution. If we're just looking very simply, those would be the compound light microscopes. But there's the transmission microscopes and the scanning electron microscopes. If you have the main difference here is basically what it does is it shoots off electrons and picks up images. But if you want to have a species that you still, or an organism that you still want to keep alive, you're going to use something called a scanning electron microscope instead of a transmission electron microscope. And like I said, that was the entire cell. Just know your organelles, look them up, be able to relate to what they're used for and how their structure is set up.